Hello and welcome to Nikolai's genetics lessons, and today we are going to talk about very interesting situation which sooner or later every one of us can face. So here's a question which I got from one of my subscribers: why do DNAs of the same length have different positions in gel electrophoresis? And here's a description of the problem. You can read it by yourself. And here's the four sequences of the same length which should appear on the gel electrophoresis as the bands of the same size, so at the same positions, but as you see, all of them appear at the different positions. While primarily factor determining DNA migration speed in a gel, like pH in this case, is its length, the other characteristics can significantly influence its movement, causing DNA molecules of the exact same length to appear at different positions. The most likely reason for such observation is a difference in the DNA molecules' conformation or secondary structure. Take a look at this picture. The most stable structure of DNA is canonical right-handed double helix, termed B-DNA. You can see it here. However, certain environments and sequence motifs favor alternative conformations, termed non-canonical secondary structures. You also can see here left-handed DNA double helix, which we call Z-DNA. Here you can see also what we call G-quadruplexes, which result of stacking of G-quartets or guanine. And here you can see hydrogen bonds, which forms between hydrogens. So this is another example of the secondary structures which are non-canonical. And such hydrogen bonds we call Hooke's-Stinn hydrogen bonds. And we can find such structures where multiple guanines goes in a row. Here's another example of the hairpins which are formed at inverted repeats, in which the stem base pairs hybridize with hydrogen bonds while the loop remains single-stranded. So here, here, and here. And here's another example of the stem and loop structure. Slipped strand mispairing at tandem repeats results in slipped structure formation like this. At the bottom of each picture you can see linear form of the DNA. You can see here motifs, and on the picture F is a depiction of homopurine-homopyrimidine sequence with mirror symmetry, which may lead to such slippage where we can get a loop like this. And this is also example of the secondary structure which may affect how DNA would move through the gel. Next, here on this picture we see example of what we call H-DNA. This is triple helix. As you see, two strands of the DNA forms are loops, where third strand of the DNA can be integrated, hybridized, and the fourth strand is going to be single-stranded. And in the last example, we see hybridization of the DNA and RNA. So as you see, though these DNA strands can be of the same length, they can take different positions when you run a gel due to possible different conformations which these DNA strands may take, which will affect the speed of movement through the gel. And this is all for today. Subscribe and see you in the next video. Goodbye.